This is Module A6. It's going to cover materials from Chapter 14 of your textbook titled The Brain. In Objective 1, we're going to identify the major regions of the brain and offer a very brief introduction to each of these regions. The nervous system is the decision and communication system for the body. The nervous system is comprised of the central nervous system as well as the peripheral nervous system, and together these two systems control every part of our daily life, from breathing and blinking to helping you memorize the facts for this exam. Sensory nerves gather information from the environment and send that information to the spinal cord, which then sends the message to the brain. It's the role of the brain to make sense of that message and fire off some type of response. Motor neurons deliver the instructions from the brain to the rest of your body. The spinal cord, made of bundles of nerves running up and down the spine, is similar to a superhighway, speeding messages to and from the brain at every second of life. When you look at the brain, I think the most noticeable region of the brain would be the cerebrum. The cerebrum is the very enlarged, convoluted area of the brain, and it's the seat of most higher mental functions, such as conscious thought, sensations, intellect, memory, as well as complex movements. The second largest region of the brain is the cerebellum. It's located in the posterior regions, and I probably should orient you. This would be the anterior, and this would be posterior. This is superior and inferior. The cerebellum tends to adjust ongoing movements by comparing arriving sensations with previously experienced sensations. Ultimately, this allows you to perform the same movements over and over again. The cerebellum is associated with regulating the coordinations of movements associated with posture and balance, for instance. The next region is the diencephalon. This is, the diencephalon is located right here. And the diencephalon includes the thalamus as well as the hypothalamus. This structure forms not only a structural but functional link between the two cerebral hemispheres and the brainstem. When we isolate the two regions of the diencephalon, we have the thalamus, and there's a left and right thalamus. The role of the thalamus is to relay processing centers for sensory information. In fact, the thalamus is a relay station for all the major sensory systems with the exception of the olfactory system, or smell. So, for instance, the thalami will relay information from the eyes, from the ears in terms of hearing, all the major sensory systems with the exception of smell. The hypothalamus, located underneath the thalamic region, makes up the floor of the diencephalon, and the hypothalamus is associated with emotion, certain autonomic functions, as well as hormone production, which will be covered at a greater extent at the start of next semester. The brainstem includes the mesencephalon, the pons, as well as the medulla oblongata. And the role of the brainstem is to relay information headed to or from the cerebrum or the cerebellum. The mesencephalon, or midbrain, processes visual and auditory information, as well as control reflexes triggered by these stimuli. So, for instance, if you were to hear a loud, unexpected noise, this is going to initiate a reflex that includes both the eyes as well as the muscles of the head turning towards that noise. These are all controlled by the midbrain. The pons, this little region right here, the pons connects the cerebellum to the brainstem. And in addition to tracts and relay centers associated with the pons, we find nuclei that deal primarily with sleep, respiration, controlling the swallowing, bladder control, hearing, equilibrium, taste and eye movements, facial expressions, and posture. So the pons has a tremendous responsibility as a controller, has a hand in controlling many of these functions. The last structure we're going to consider is the medulla oblongata. This is the inferior-most region of the brain. And the medulla is continuous with the spinal cord. The spinal cord will continue off the medulla as it travels down the vertebral foramen of the vertebrae. And the medulla its main role is to relay sensory information to the thalamus and other centers and other regions of the brainstem. The medulla contains centers that regulate autonomic functions such as heart rate and blood pressure, as well as functions such as digestion. I know that that was a lot of information about these various regions of the brain. We're going to cover each of these more specifically as we move through this module. I just kind of wanted to give you a taste of some of the functions. I think it would probably be very beneficial for you to try to orient yourself in the different regions of the brain in terms of their location. What we have is the cerebrum is going to be, this very large region that most of us could identify as a brain, and that coupled with the cerebellum make up the majority of the brain mass. If we were to open this up and look in the central region of the brain, this is where we're going to find the diencephalon, the mesencephalon, pons, and medulla. So this is kind of the brain stem and the midbrain region, and this is all again nestled right here in the middle of the cerebrum and just anterior to the cerebellum. So just trying to orient you to some of the structural components of the brain will help us later on as we start looking at these regions in more detail. So don't focus so much about the various functions or regions associated with these areas, but try to orient yourself to the orientation, and we'll cover the specifics later. We find that the brain is large, it's very delicate, and it contains the neural tissue discussed, both the neurons as well as the neuroglial cells. The brain contains internal passageways and chambers filled with a fluid known as cerebral spinal fluid. And we'll talk more about the composition and the function of cerebral spinal fluid later in this module, CSF. We mentioned the 6 major regions of the brain and noted that each have specific functions. And we find that as we ascend from the medulla up towards the cerebrum, the brain functions become more and more complex. And this is a pattern that we're going to see and discuss a bit later. At the pinnacle of that complexness, we find conscious thought and intelligence And these are all going to be found in the cerebrum itself, or the cerebral hemispheres. In Objective 2, we're going to look at the cerebral spinal fluid, along with the meninges as well as the blood-brain barrier. The brain itself is, as noted, a very delicate organ, and it requires physical as well as biochemical protection. In terms of physical protection, we have the bones of the cranium surrounding and protecting the brain. We also have layers of membranes known as the cranial meninges, as well as a fluid that circulates around the brain, serving as a shock absorber and help cushioning the brain. And this is known as the cerebral spinal fluid. In terms of the biochemical isolation or biochemical protection, we have the production of a blood-brain barrier, and we'll discuss its implications in a minute. The cranial meninges, there are three layers. We have the dura mater, arachnoid mater, and pia mater. And if we look at this diagram, we can identify, here's the dura mater, this is the outermost layer, and as we work and become more deep, we would find the arachnoid mater as well as the pia mater. This is another view. Again, we have the dura mater right here, followed by the arachnoid mater and pia mater, the three layers of the meninges. And the three layers of the meninges are going to surround the brain, as well as continuous with the spinal meninges as they surround the spinal cord. Dura mater translates into tough mother. This is a very tough membrane surrounding, it's the outermost meningeal layer. It has an inner fibrous layer and this outer fibrous layer that's fused with the periosteum of the various bones that make up the cranial vault. So if you stop and try to think about these bones, you would have the frontal and the parietals and the occipital, the temporal bones, and then as you move more, I guess, inferiorly, you would have the sphenoid and other bones making up the cranial vault. The arachnoid mater covers the brain. It's intermediate between the three maters. And we find that there is a space created underneath the arachnoid mater called the subarachnoid space. So we can kind of start layering this. We have this outer pia mater that is continuous with the periosteum of bones that make up the cranial vault. We have the arachnoid mater, and then we have a pia mater. We can identify several spaces that lie between these maters, and we've already looked at the subarachnoid. So subarachnoid would be the space between the arachnoid mater and the pia mater. Lastly, the pia mater is actually attached to the surface of the brain, and it's held in place by astrocytes. We didn't discuss the attachment as a function of the astrocytes earlier, but it is held in place. Just I'll throw in this last space, the space between the dura mater and the arachnoid mater. This is known as the subdural mater, or subdural space. And in the text, let's see if I can find the page really quickly. There is a clinical note on page 466 that look at that covers subdural hemorrhages. And those would be hemorrhages occurring in that particular space. Parts of, we find in certain regions of the brain these very deep fissures. And we find that the dura mater continues down in these deep fissures, and these are known as dural folds. So they're folded inner layers of dura mater extending into the cranial cavity. And the role of these folds is to help support and stabilize the brain. And we can actually identify, we've given anatomical names to certain regions. We have the falx cerebri. This is a very large dural fold that extends down into the sagittal fissure, something we'll learn in lab. We have the tentorium cerebelli. This is a fold right here that helps stabilize the cerebellum. And then we have the falx cerebelli right here. And again, we're going to look at these structures in the lab. I just wanted to mention them now that we were, while we were discussing the meningeal layers. Cerebral spinal fluid surrounds all of the surfaces, or the exposed surfaces, of the central nervous system. And there's an interchange between the fluids of the interstitial fluid with the cerebral spinal fluid. Functions of CSF include the cushioning delicate neural structures, supports the brain, and this is also where you have the transport of nutrients, waste products, as well as certain chemical messengers associated. So it sort of serves a similar function of that of blood, in which you have the transport of nutrients and waste products. Cerebral spinal fluid is produced in certain regions by structures known as the choroid plexes. And the choroid plexes, they're specialized ependymal cells associated with a capillary bed. If you remember, the ependymal cells are the neuroglial cells that line the ventricular system. These choroid plexes are going to secrete CSF into the ventricles. They remove waste products from the CSF. And they're also used to adjust the composition of the cerebral spinal fluid. About 500 milliliters of CSF are produced each day, and about that same amount would be drained back into venous circulation. So it's going to be drained back into a vein. If you were to try to detect the amount of CSF present at any given time, you'd find about 150 milliliters present. So it's constantly being produced, it's circulating, and then it's constantly being reabsorbed into a vein. To look at the movement of the CSF, we see that it's produced here at the choroid plexus, and there are several regions. Here's a choroid plexus. We also have choroid plexus here. And it's going to circulate from those plexes through the ventricles, ultimately to the central canal of the spinal cord, down that canal, and into the subarachnoid space that then goes up and around the brain, spinal cord, as well as the cauda equina. This is something that will be introduced in lab. So just follow the little red arrows as it moves through the body, through the ventricular system of the brain, down through the spinal cord, the central canal of the spinal cord, and then finally up through the subarachnoid space and around the brain. When we look at the subarachnoid space, this is also the region where the cerebral spinal fluid, returns to venous circulation. Here is the subarachnoid space. By the way, this was arachnoid. Many of you would associate that with spiders. And this particular structure was named that because of the spider web-like appearance of the fibers within this subarachnoid space. FYI. Within the subarachnoid space, we find arachnoid villi. And you should associate villi with some type of little projection, some type of little finger-like projection. And these are extensions of the subarachnoid space. They extend through the dura mater, here's your dura mater, and into an area that is part of the superior sagittal sinus. And again, that's all of this blue right here. So the villi is going to extend up through the dura mater and into the superior sagittal sinus. And a sinus is an area that, this particular sinus is an area where veins kind of open up and blood is free to circulate within the sinus before it returns to another vessel. So this is where the CSF is returned to venous circulation or returned to a vein. You can see that as the CSF flows through the subarachnoid space, it's then able to maneuver up into these arachnoid villi, and then it can be deposited or lost back into venous circulation. Arachnoid granulations are large clusters of these villi, and again, its role is to absorb the CSF into the venous circulation. If 500 milliliters are being produced each day, 500 need to be reabsorbed or absorbed back into venous circulation. There are many clinical implications for a blockage of this, this process. For instance, if 500 milliliters are being produced and you do not have the absorption of the CSF back into venous circulation, you end up with an increased pressure in the brain. In infants, this can lead to hydrocephaly. This is water on the brain, and there are implications in adulthood as well. This is just putting those two figures together. Here, again, we've seen this cross-section of the brain, this sagittal section showing the movement of the cerebral spinal fluid indicated by all these little red arrows. Where is it produced? Where is CSF produced? Again, the choroid plexus, and we're going to find the choroid plexus associated with the lateral ventricles, the third ventricle, as well as the fourth ventricle. And if we haven't covered these structures in the lab yet, we will. But for now, just kind of note where the CSF is being produced. And then it's going to flow through the ventricular system, which is continuous with the central canal of the spinal cord. And then it's going to flow up and around the subarachnoid space. Within the subarachnoid space, the CSF will then be resorbed by, or through these villi into these sinuses, and then return to the blood, return to venous circulation. The blood supply to the brain. The brain is a very important organ, and it has a very intricate blood supply. This blood supply is going to bring nutrients and oxygen to the brain. And we find that the CNF is isolated, at least the neurologic tissue is isolated from general circulation. Between the endothelial cells of the capillaries of the CNS, in fact, you're not really familiar with this terminology, so I'm just going to kind of remove it right now. What I'd like you to know is that astrocytes control the blood-brain barrier, and they do so by releasing chemicals that controls the permeability of the blood vessel. The blood it can quickly be altered in terms of composition, and it's very free to hormones. And the brain cannot deal with that change of blood composition. So a blood-brain barrier is required, and it's produced by the astrocytes, and that is how the brain is still able to benefit from the nutrients and the oxygen content of the blood, yet much of the other constituents of the blood is isolated. There are 4 breaks in the blood-brain barrier. We have a break in the hypothalamus, the posterior lobe of the pituitary, the pineal gland, as well as the choroid plexus. And most of these, at least the first 3 of these breaks, are associated with the endocrine system, with hormones. Hormones are produced by these various glands, and they need to be dumped into circulation. So that is 3 of the 4 breaks. The choroid plexus is where the ependymal cells help with the production or maintenance of the cerebral spinal fluid. So let's just kind of review. The meninges are going to stabilize the brain in the cranial cavity. The CSF is going to protect and cushion the brain against sudden movement. It's going to provide nutrients, and it's going to remove waste products produced by cells. And we have the blood-brain barrier and the blood-CSF barrier that selectively isolate the brain from chemicals in the blood that might disrupt any type of neurologic function.